Okay. Can you guys see the screen? All right, perfect.
Yes.
So the first presentation will be, if it loads, here by Celine Carly from DKFZ. She will talk to us about autophagy pathway and small extracellular vesicles in connection with irradiation. So Celine, if you are ready to share your screen, and you have 15 minutes to talk about your project.
Yes, hi, can you hear me?
Yes, we can.
Okay, perfect, and I will try to, you cannot start screen sharing. Yes. Okay, maybe, and it should be now possible. Can you see my presentation?
Yes, we can.
Okay, perfect. Then I would just start right away. Okay, so hello, my name is Celine. I'm currently a PhD student here at the HITR facility in Heidelberg. I did my master thesis in radiobiology, and since a talk in radiobiology was wanted, I think I will do this on my thesis. So I did my master thesis at the KFZ and Professor João Zeco's group. We cooperated with the HIT facility because we irradiated ourselves with carbon ions. This one was my wonderful supervisor, Dr. Geraldine Genard. She's a biologist and I was the physicist. So I was really working on the edge between physics and biology. And yes, so be kind with your biology questions. I will do my best. So my topic was the effects of carbon ion and X-ray radiation on the biogenesis of small extracellular vesicles with focus on the autophagy pathway. Don't be scared of this lengthy title. I hope I can explain it now in some detail. So from this, these were my three main topics: the irradiation, the autophagy pathway, and small extracellular vesicles, which are abbreviated by SEV. But what are these SEVs? SEV are tiny vesicles that are emitted by every cell. Their formation starts with the endocytosis, and within these small vesicles within the cell, even smaller vesicles are produced, as you can see here. These are then loaded with certain proteins, DNA or RNA. So they are very cell-specific and loaded with the cell content. When these then fuse with the membrane, the SEV are released into the extracellular environment. There they can connect with other cells and induce certain reactions. So they are a main part of the cell-cell communication. It was already shown that they participate in tumor growth, metastasis formation, and immune suppression. And this is what makes them so important for cancer research, because they play such a huge part. The other biology factor was the autophagy pathway. This is the autophagy pathway in its full beauty, but we can just for the first step focus on the main part. Maybe some of you know this pathway as programmed cell death, but it's even more than that. You can see here that after the initialization, a membrane is formed. This membrane will result in a vesicle, and this vesicle is loaded with waste. So everything that is damaged in a cell can be put in these autophagosomes. When these then fuse with lysosomes, the waste gets degraded because these lysosomes are acidic. So it's basically to get rid of all the waste within the cell. So it's actually for maintaining the cellular health, because you want to get rid of the waste, which is, from especially and then induced by irradiation. The irradiation, as I said, which was in my title, we have X-ray and carbon iron irradiation. Why we did both is because it was shown, especially in vivo, that carbon irradiation induces a different immune response in animals. And we want to see if this also happens within the cells. So our studies were mainly in vitro on cells. These three parts, the irradiation, autophagy, and the SEV, are connected. So irradiation is said to enhance the biogenesis of SEV. So when a cell is irradiated, it starts to emit more SEV to connect to other cells. Due to the cellular stress and the damage in organelles, it also activates the autophagy pathway, but not only as programmed cell death, as I said, but to maintain the cellular health. Recent studies have found that these both pathways, the SEV production and the autophagy pathway, are tightly connected. Some of the proteins from the autophagy pathway were found on SEV, so there must be a kind of a connection. So our question was now, how do these three interact? What does the irradiation has to do with the interconnection of the SEV pathway and the autophagy? So to sum up, the SEV are a main part in the cell-cell communication and are really, are really important for cancer progression. Since this is connected to the autophagy pathway and triggered by irradiation, we block the autophagy pathway to examine the SEV released after irradiation. So to show you this a little bit more in detail and what I've really done is here my project outline. So the first step was to actually inhibit the autophagy pathway within the cells. Afterwards, when we have done this, we irradiate the cells and collect their SEV. The last step was to do a spheroid experiment, which I will explain at the end. So first, we have to go back to all these proteins here. As I said, there's a connection between the autophagy pathway and the SEV. Some of these proteins here and here were found on SEV. So we decided to block the autophagy on two different ways. The first one is here, directly at the beginning with 3-methyladenine, here abbreviated by 3-MA. And the other one was chloroquine, which blocks the part here. We did this to see if there is like an important step here in between, which is needed for the SEV production, so that we can at the end see, okay, maybe the step beforehand or the step afterwards is important for the SEV production. Oh, sorry, that was backwards. This way. As it is for every other drug, when you apply it to cells, when you pass a certain threshold, the cells can die because it's a drug. So we performed several cell viability assays to ensure that our cells are, they are still living when we irradiate them. The first one was a clonogenic assay. Probably some of you will know this. With this, you can track the survival fraction. So you apply the drug to the medium and see how many cells can actually form colonies. And colonies are defined just as these cell clumps you can see here, which contain more than 50 cells. And with this, we can see what dosage the cells still survive. The second one is called the cell tidal flow. I'm very sorry for this image. This is actually from the company itself. Don't know why it's so pixely, but it actually measures the ATP content within the cell. So the ATP is responsible, it delivers the energy for the cell. And when the cell is still metabolically active, it induces a reaction that produces light in the end, which we can detect. And so with this assay, we can see if they are still metabolically active. The last one was protein extractions and Western blots. As you may remember from the plot from the autophagy pathway, there were a bunch of proteins active in this process. And when we extract these from the cells and plot these on the membrane, we can actually make them visible. And this is what you can see here on the membrane. And then we can follow with increasing dosage from the drugs, for example, which protein is active and which is not, to see if the cell is dying or if the autophagy is just inhibited and it's still alive. So when we have fixed this and our cells are still alive, we were happy and we can finally start with the irradiation. We had in total 9 different conditions. Surely you have to make a control of everything to see if it's really an effect of the certain modality or not. So we had the X-ray irradiation, the carbon ion irradiation, but also the chloroquine treatment and the 3-MA treatment. So a lot of cells to prepare. And then we irradiated the cells. As I said, we did this with X-rays, but also with carbon ions. And you can see here the nozzle from the experimental area at the HIT, where we irradiated our flasks here. The cells were detached here on this side of the flask. As you can see, the medium's on the bottom, so we really have to be fast to not induce further cell death in our cells. And this medium contains then the SEV because, as I said, the cell just admits them into the extracellular environment. And so after several days, we collect this medium. After several centrifugation steps, we then had our SEV samples. I will not go into detail. It's just to get rid of all the cells which are also in the medium, all the membranes, and just to have in the end a sample which only contains the SEV. These samples we then examined for several properties. first we did a lot of Western plots, again, with these proteins where we can see, okay, what is on the SEV or not. But the most interesting for me was the confocal microscope. As you can see here, you have this pretty colorful images with the blue ones representing the nuclei of the cells. And when they are here a little bit reddish, it means they were indeed transfected. You can see here maybe these bright red dots. These were the tiny vesicles within the cells where the SEV are. So we can see how the drugs affect the SEV production directly in the cells. And this was really interesting. And this is one of my results. I just have to move it a bit. So here, and what directly pops up, first of all, this was here the non-treated group. So there was no drug applied. Here was the chloroquine drug, which inhibits the end of the autophagy pathway. And this is the 3MA, which blocks the beginning of the autophagy pathway. And what you can directly see, no matter which irradiation modality was chosen, the number of SEV within the cells was dramatically decreased. Like you can see here, it's nearly 0, and for all the other groups, it's much higher. So there was the first thing we see, okay, there's actually something happening, and there could be the connection. So we started another huge experiment, which were the spheroids. Spheroids are tiny little tumors, so you just clump a bunch of tumor cells together, put them into a matrix, and see what happens over the time. Why we did this? We put the SEV from like the different treatment and irradiation groups, extract them there, and put them on the spheroids, because the SEV are said to be active during metastasis formation and invasiveness. So we want to directly check if there is something happening, and to see like the indirect effect of the SEV on other cell culture cells in this type, the spheroids. This is how it looks over time. Like on the first day, you really have this circular shape of the spheroid, and then it starts to spread through the matrix, and you can really see it's getting arms. And yeah, unfortunately, I cannot show you any results because the experiments are still ongoing. They are quite lengthy to extract this process and to build the spheroids. So, but I really hope that during the next half year, there may be something interesting coming out of this research. So yeah, the goal of my thesis was basically to see the difference in the radiation modalities on the SEV, and to reveal the connection between the SEV pathway and the autophagy. There's still a lot of work to be done. I would never say that it's now finished. It's certainly not, but I hope I could show you a little bit the connection between irradiation, autophagy, and SEV. So thank you very much for your attention, and I hope you liked it.
Thank you, Celine. So we still have a little bit of time for questions if anyone has something to ask. Okay, Sharnay?
Hi, can you hear me?
Yes, I can hear you.
Hi, Celine. That was very interesting. Thank you. I just wanted to know whether or not you characterized what is actually inside of your SEVs.
You mean what is within, like contained in the SEV? That is work which was actually done before that. SEV are a very heterogeneous group. There's a lot of stuff inside them. There can be RNA, there can be DNA. It's like, yeah, basically what the cell wants to deliver to the other group. So that's quite heterogeneous. You cannot say like, it's an SEV because it only contains DNA. It's not like that. It's basically defined by size. So it's between, I guess, 200 nanometers and something like that, when I remember the numbers correctly. And this is how you define them, and by outer surface markers. So you define them by outer surface and by the size, and not really by the content.
Thank you.
You're welcome.
Okay, we have a question from Coyote in the chat. Please, can you say something about the radiation dosimetry, i.e. how you were able to verify the dose at the positions where the cells are placed?
Yes, so for the X-ray machine, I didn't show you a picture. It's the multi-rad 225. It's yeah, basically you have like a shelf where you put your cells on it, and this machine is very, very user friendly. It basically does the dosimetry on its own. Like it has a small dosimeter below the cells and then verifies that you applied the right dose. Surely we did in advance some dosimetric measurements with former chambers to verify if we really apply the right dose and the right position. But yeah, it's a really user-friendly device. At the HIT facility, normally the HIT staff themselves do the QA, but we followed them around. They have also different measurements with ionization chambers from PTW, where they checked that the Bragg peak is in the right position, and this position is marked by a laser. So it's really easy to just align it with the laser, and then everything should be fine like normally. But we checked that in advance with the HIT stuff and with dosimeter.
Okay, just one last question. What type of cells have you worked on from Domenico?
We have worked on a KPC cell line. That's a mouse pediatric cancer cell line. So it's pediatric cancer from mouse cell lines, and also on the PANC01.
Okay, great. So I guess we can move on to the next presentations. Thank you so much, Celine. That was really interesting.
You're welcome.
So the next presentation is from Adam Steinberg, jointly from University of Melbourne and Manchester. So Adam, if you can share your screen, and the floor is yours.
Great. There we are. I'll start the timer too, so hopefully I don't go over time. Hi everyone.
Perfect.
Yeah, hi everyone. So I'm Adam. There are various details there on the screen. In the bottom corner is a link to get these slides if you want them. If you don't catch it now, it'll also be at the end to see again. There's also my email and Twitter for the usual stuff for that. Today I'm going to talk to you about a project we're doing at the University of Melbourne, which here I've titled Enabling Rapid Depth Scanning for Charged Particle Therapy, as I'm sure you read a moment ago. And hopefully by the end, you'll know what I mean by that and what we're doing towards it. And just so you know, because this is obviously a Zoom session, over in the top right corner is a protected region. So if you make the screen of me like small and put it over there, nothing will be behind that. So you're safe to put my face in that corner, or just maybe disappear entirely. I'm not going to have that many interesting expressions, but there'll be some stuff you want to see. So in the next 15 minutes, I'm going to be talking to you. Well, think about the title of the session. We'll start off with the dream, and after the dream, of course, comes the beam. And then I'll talk a bit about the turbo project, the thing we're talking about here, and then a bit of time for questions. But of course, any dream has to have a dream team. I can't take credit for all the stuff we're doing. So here's just kind of everyone involved. Our kind of academic leads are Rob and Susie there, and just into the postdoc. Hannah and I are both second year PhD students, and we're both in this joint scheme between Manchester and Melbourne. I'm currently in Melbourne. It's quite dark outside. I'm envious of everyone who's been eating lunch every day, because for me, it's been quite a late dinner. So let's talk a bit about the dream. From a medical perspective, a bottleneck in the treatment of cancers for particle therapy is the time to change the depth of the Bragg peak, the energy layer switching time, you might call it. What am I talking about? Well, if you look at these two example peaks here, if you have to switch the depth from the furthest to the closer one, it takes time to switch the beam energy from, say, 230 to 150 MeV. So you have to ask a question. Can we vary the beam energy fast enough to remove this bottleneck so our treatments aren't limited by the time to switch the energy or the energy layer? In order to do this, our accelerator has to have various capabilities I'm not going to discuss so much, but also the beam delivery system has to be able to cope with these rapid changes. I'm going to talk more about the beam delivery system today. I'm not going to be talking about those personalized range modulators with the spikes that we've been seeing a few times. We're talking about something that can still do potentially scanning while still changing the layers through the beam energy itself. Now, what does this mean from an accelerator perspective? Well, we might remember from some of the talks about accelerators that we're going to need stronger magnetic fields to steer higher energy beams. You might be wondering, why would this be? Well, think of an analogy. Imagine you're standing in a corner, that's you, by the way, and you're going to pull your friend towards yourself. First of all, you've got a small friend, and you pull them towards you as they run past, and they run past kind of slowly. And you can see that you managed to get them quite a lot of turning. Then you've got a bigger friend, and your bigger friend's going to run faster. As your bigger friend runs faster, you pull them with the same amount of force, and yeah, they still have some bending, still some turning, but not nearly as much. Think of your heavy friend, who could be traveling a lot faster as well, as the higher energy particle or the heavier particle, so carbon rather than proton, and your smaller friend, who's traveling slower, as a lower energy particle. Higher energy friend needs more bending, high energy particle needs more bending. Now, in the synchrotrons, which we've heard a lot of talk about that for gantries and this magnet ramping machines, the magnetic fields increase with time. So as the beam energy increases, the magnets increase their fields. The question is, can we deliver multiple energies without ramping up these magnetic fields? And the trick we're going to employ is to vary the magnetic fields in space rather than in time. So rather than increasing the magnetic field, over the course of seconds even, we're going to space out higher and higher magnetic fields to get our beam to be turning the same amount regardless of energy. Now, this isn't trivial, so we're going to need some sort of demonstrator machine to show this can work. Let's talk about the facilities we have here in Melbourne. We have a pelletron accelerator, which has been mentioned very, very briefly towards the start of the course, I think. But just in case anyone can't remember, it's an electrostatic DC accelerator, so it only goes to relatively low energies. It can go up to 3.5 MeV for protons. It can also do kind of helium-1+ ions and some other things. I'm not sure about the energy ranges for those. And it has 3 beam lines. I say it rotates between the beam lines. What do I mean this accelerator rotates? Can you imagine the Large Hadron Collider rotating? But we're not talking about something like the Large Hadron Collider. The pelletron is this machine over here. Up above it is a whole big stack. That's where the acceleration happens. The particle starts at the top, goes down to the bottom, then is turned into the horizontal plane. And then over here in the corner, there are 3 beam lines, which can each get beamed separately. Just to make it clear what I mean by that, this is a rough schematic of the room. We've got a blue, red, and green beam line, handily named blue, red, and green. And the pelletron can rotate between these 3 points. Our proposal is to build a new beam line, maybe here, maybe here, maybe here, we're not sure yet, to demonstrate some of our ideas. So what are our ideas? What are our plans? Well, I should really tell you what turbo stands for. As you see there, turbo is what we call a very nice acronym. It stands for Technology for Ultra-Rapid Beam Operation. Also stands for something you can easily pronounce and remember. Now, our beam line, schematically, beam starts over here from the pelletron, ends down here at some sort of beam stop or whatever. Obviously, stuff happens in between. We're going to need to have some sort of matching sections to get the beam from the pelletron to match whatever optics we're going to have in the middle. We have some energy degrader. We're trying to show that the energy can be varied rapidly. The pelletron varies relatively slowly, so we need to vary the beam energy much faster with some sort of energy degrader, a bit like a cyclotron has. We're going to need some sort of achromatic arc where it can transport lots of different energies. Say this one at the top is a high energy, this one at the bottom is a low energy, can transport them all simultaneously. May there be some sort of dispersion in between where it goes out and then back in. And to be clear, this has to be bent as well. This is trivial if it's a straight line, because everything just goes in straight lines. But if your arc is bent, it's actually quite difficult to get all your energies to go through together. Finally, we're hoping to have some sort of scanning section and then do some sort of dosimetry or treatment-related things at the end before that very final beam stop. This is just a schematic. It will take a lot of time to get to all these parts. So I'm going to tell you a bit about what we've been doing towards this. We're at a relatively early stage, so we're still designing, say, the magnets to go in the achromatic arc. Because we're going to use static magnetic fields, we can use permanent magnet blocks to produce the desired fields. To make life easy, we're going to make 3D printed holders, because that means you can prototype things quickly and develop things quickly and swap out one thing for another thing, and it can take not very long at all. So here's just a quick video to show those are magnets that slot into the holder. Here they're going to do some gymnastics because they can. And you can see at the bottom, you've got those arrows, and those arrows say which way the magnetization vector should go. So should my magnets be this way around or that way around? And they just slot in nice and easy. And the idea would be that this produces some magnetic field in the middle, which is the pattern you like. And I'm happy to say that the initial prototyping looks positive. Oops, that's going to show again. The initial prototyping looks positive. And how can I say that? Well, anyone from the UK might recognize the phrase, here's one I made earlier. So here's one I made earlier. And you can see it's not quite the same arrangement as we have in the picture there, just based on this number of magnets we had available. This is also backwards, so you can see the arrows possibly just there. And what we've done is we've measured the magnetic field produced by this. That's on this slide. And I won't belabor the point because it's not very exciting necessarily, but this is meant to be a dipole magnet. So the field goes from the bottom to the top. That's from here to up there. And what we can see is that, yeah, in the middle, the field does pretty much go from the bottom to the top as we'd expect. So in the future, we could put beam through going straight towards this green there, and then we would be bent the right amount. So that's looking pretty positive so far. We're also planning to do beam profile measurements. So what does the actual beam coming from the pelletron look like? We've got a fiber scanner for beam measurements. What do I mean by a fiber scanner? Well, the beam is going to come along in this direction through the beam pipe towards this funny looking box here. And in this box, there is a fiber, which funny enough scans. It scans up and down in this plane. And it does that because it's an optical fiber. It scintillates, it lets off some light. That light then goes down this tube, down to a computer somewhere, and you can measure the signal. Using that, you can translate it to a beam intensity. We've done that a few times, and what we can see is, oh yeah, there's a lot of noise here, not entirely sure where the noise is from, but these beam profiles definitely look like it could be a real particle beam. So first measurements are what you might call promising. Oops. Now in the future, we're planning to do some more detailed measurements for accelerated people. It's a slit grid experiment, where what you do, and I'm not going to talk about it for too much time, but you select one point in position far up the beam line. You then let the beam kind of diverge out so you get the angular information further down. This will measure with the fiber scanner. And by measuring the angular information here and position there, then move the position a tiny bit, measure it again, then move this thing a tiny bit and measure it again. You can get a detailed information about the phase space, the position versus angle correlation at that point. Once you know this, you know everything about your beam and you're good to go. This is a future measurement we've got to do, but actually we've already got our slits ready and existing. This is just so tantalum sheets inside the metal. And if you're wondering about the length scale, again, here's one I made earlier. In fact, it's the same one I made earlier. And it's about this big against my hand. So it's pretty small to go inside the beam pipe. It's great having all these props. And that's actually all I have time to discuss. I'll just kind of give you a quick overview of the various things. So I've not got time to discuss a whole range of other stuff to do with this project. There's the energy degrader. There's the matching section. There's the beam scanning system. There's all the other diagnostics other than stuff I just discussed. There's all the internal pelletron features, which are all really interesting. There's all the beam control stuff, a bit like the talk we just had. There's all the corrector magnets, because no magnets are perfect. And of course, there's also the full energy implementation. This is a low energy demonstrator, 3.5 MeV protons is the maximum. In a real clinic, you have 250 MeV protons. This is only a demonstrator, and hopefully you'll have a scale-up version in the future. So that's been a real whirlwind tour of a pretty large project. What do I want you to remember? Well, we're working on this large energy acceptance beamline at the University of Melbourne that we're going to do experiments on. The idea is that I can demonstrate the requirements for rapid energy layer switching in clinical systems to remove that bottleneck. And also the design work so far, including the magnets and diagnostics that I've discussed, but also all the other things are going pretty well, but obviously there's plenty more to do. And so with that, I'll leave you with a final image of the pelletron controls. This is a machine from the 1970s, and it's all these manual knobs and levers, and operating is like magic that I don't understand. But that's all I have to say. Thanks, everyone. Again, here are the details for the slides and my email if you want them. But if you've got any questions now, I'd be happy to hear them.
Thank you so much, Adam. That was a very interesting and cool presentation. So if you have any questions. Okay. So I don't see anything in the chat. Any questions for Adam? He's gonna be more than happy to answer.
Can I ask? I'm not a student,
Yeah,
as you know. Why are
course,
you
Professor.
doing this?
So I can give a few different possible answers. Some sound more pessimistic than others. Maybe the most optimistic one is to say, for treatments where the beam energy needs to be very rapidly, both to remove that bottleneck, but also for something like flash therapy, current gantries and current beam transport lines can't get a wide range of energies from the accelerator to the patient rapidly enough. If we're talking about, say, 100 milliseconds, or even longer timescales than that, ramping magnets just isn't going to do. What we've got here is an accelerator with fixed fields. People might remember in the gantries talk earlier today, they mentioned these fixed field accelerators. This is an example of one of those. I just didn't give them those names because the name's a bit, people don't like it. By using these fixed fields, you remove all these time constraints and you have much more freedom to do other cool stuff. There's still complexity. Getting a wide range of energies through at the same time isn't easy. If it were easy, everyone would be doing it. But if you can do it, it has the potential to make treatment patterns available which just aren't possible with current systems. Also because it's fun.
Okay, thank you, Adam. We have a question in the chat. So from Felix, will you change the Pelletron settings to switch energies or use the energy degraders or both?
So I invite you again to look at this front control panel for the Pelletron. It's a bit of a mess. Switching energies to the Pelletron is a slow process. I actually have no idea how it's done. The technician comes in the mornings at like 6 a.m. and sets everything up. So I have an idea how some of these knobs work, but not for most of them. So no, if you tried to change it with the Pelletron itself, it would be quite a long timescale for the changes. The energy degraders are what will allow us to change the energy much more rapidly on the timescale of seconds or faster. I'm not designing the system, so I can't speak in detail. But no, it should mostly be down to the degraders rather than the Pelletron. Pelletron will be static at probably 3.5 MeV and then degrading down from there.
Okay, Florian, maybe for the last question.
Yes, sorry, I'm not sure if you already mentioned it, but you're talking about a large energy acceptance. What do you have any simulation or ideas how big this energy acceptance should be, or?
Yeah, so lots of ways I could answer that. Maybe this one's a comparison. So for a lot of GAN trees and beam transport lines we tend to see, the momentum acceptance, which isn't quite the same as energy acceptance, but it's a bit more natural units to use. The momentum acceptance is plus minus like maximum 1%. 1% is quite a big momentum acceptance for these things. For this sort of beam line, we're looking at more like plus minus 40%. So order of magnitude higher and more. So it's a very different type of problem. I showed the picture earlier of the beams diverging out and then coming back in. If you showed this to a synchrotron person or to a normal gantry person, they'd say that's crazy, that'll never work. And maybe they're right, but also it probably will work. So yeah, but yeah, plus minus 40% in momentum and in energy, probably about 0.5 MeV to 3.5 MeV protons. But I'd say that the momentum's a bit more natural units to use.
Okay, thank you.
Thanks.
Thank you, Adam, for a very interesting presentation. So once again, if you guys have any other questions, you can directly message him. Our next speaker is Chris Taps, jointly from CERN and Riga Technical University. So Chris Taps, the floor is yours if you want to share your screen.
Okay, everyone sees my screen, and is it changing? Yes, great. Okay, so I actually structured the presentation quite similarly. And I really went with the name because it's a really good name, Beams and Dreams. Yes, it was Manjit's idea because she's saying give the credit. So we're giving the credit where it's due. It's a good name. We have to give her that. So a little bit about me back. So yeah, I'm a first year PhD student. So I'm just still at the beginning, and I'm also a doctoral student at CERN. Originally from Latvia, and my previous background is more or less non-academia related. It's medical physics engineer in a clinic for 4 years, and also like just being an engineer in industry for medical devices. But going back to the real theme for today, the beams and dreams. So I've actually will try to cover two topics: beams, or my beam, which is heavy ion flash, and dreams, which in our case is a particle therapy center in the Baltics. We'll start with the beam. I have to say that sadly, currently my beam is theoretical. It's a beam in Monte Carlo for now, but I hope we move in the future because we need to move also to experimental part. So I'll just quickly outline where we are now with the theoretical framework for all that. So yeah, it all started as a summer student project last year at CERN, and currently I'm working in that as a part of my thesis project together with João Seco from DFKZ and also Maurizio and Mariusz from CERN. Well, Mariusz is not CERN now, he's more CEAST and PSI, but CERN is where we met. And the main thing that we are trying, sort of the idea behind it all, the heavy ion flashes, could we and should we do heavy ion flash, because it's a bit under debate, because mostly, I mean, all the flash experiments mostly have been done with electrons, a bit with protons, but mostly electrons. So the thing is also to check a bit, of course, a good mechanism explaining it all is still required, but it's a good start to just look if we should go into heavy ion flash, sort of from the perspective that, of course, then we need a bit harder accelerators. So let's check if we really should do that. How we are doing it, the idea is just to consider different ion types, do the Monte Carlo simulations in GN4, do the biologically effective dose distribution optimizations, and then from that we get LET distributions, which we correlate into flash effectiveness as potential flash effectiveness, of course. So what are we looking at from ion type? It's of course the ones that are in the clinic. It's protons and carbon. Then there's a huge debate for helium, and I will outline later. And we're checking helium-3 and helium-4. And then we sort of said, let's check everything in between that. So we took lithium, beryllium, boron. Of course, they're ones that are not really used for therapy, but let's just check it anyways. And also nitrogen and oxygen that go a bit over carbon, and those are actually considered for therapy. So what we do, we simulate over 350 energies, sort of to simulate the range where what we would need in the clinical beam, 5 to 35 centimeters, and score all the mixed beam parameters, and we score them individually, of course, by the stable isotope type, because all the parameters in the further calculations need to be defined for each of the particle types. We use quite an easy beam, just a point like pencil beam. We add an energy spread, so it's a bit more realistic, and we score it just by integrating the depth dose in the whole radiuses. But currently I'm exploring like sort of pencil beam kernel approaches, because we need to do profiles later on as well. So the main idea is that since flash protection has been mainly witnessed with the threshold dose, which appears to be around 8 to 10 grays, the idea is that also this threshold is more based on the physical dose, because from our perspective, the thing that we are investigating is a physical mechanism. So the idea is we do biologically optimized uniform SOBPs with the 10 gray physical entrance dose. Of course, in water, not in a human body, simple water geometry. We calculate the LET maps after we optimized, and we transform the LET maps into solvated electron production yield. Solvated electrons, why them? It's, of course, an idea from my supervisor João. It's that basically, since it's one of the first products that appear in the water radiolysis project, in water radiolysis, when it happens, it's one of the first particles that sort of are there in the process. So it could be the mediator for flash effect. So a small peek into the results, what we got is just a bunch of optimized bioeffective dose distributions. We transformed them by LET to the normalized solvated electron production yields. And what we get in the end are the sort of estimates for flash effect corrected dose. And the idea here that we of course see is that actually from this, it looks as we go heavier, actually we lose possibly the normal tissue flash protection effectiveness gets worse. So the thing is that maybe, and of course we're looking more into that and we need to validate it by experiments, is that heavy ion flash with the normal convention of flash that it protects normal tissues, it might not work. It would be better with lighter particles, but of course we need to validate all that. And also, of course, there are a lot of just the next steps also in the physical framework for all that. by, like I said, changing the IDD approach to pencil beam-like convolution, and also we should investigate a bit more the soul-weighted electron production yields by doing the simulations ourselves, of course, and also do some comparisons with another modality that later guys will do, because also we need to just see how we would compare in flash world with the very high energy electrons. Also, there are some takeaways, of course, along the way, because there's a lot of things that I get from these data. So it's also just interesting things on the VHEE that I already simulated, the crazy field size dependence, and just thoughts on the RBE. Also, the target fragmentation effect on the entrance RBE, like the one that was mentioned today. And for us, sort of smoothly transitioning into the next section, is actually the battle, I would say, between helium-3 and helium-4. Helium-4 is the one that's mainly considered for clinical use. There are some studies which are like sort of interesting. Could we do helium-3 because it's a bit easier from accelerated perspective? It's doesn't seem that helium-3 would be better for therapy. It's like when we do look at physical dose distributions, yes, helium-3 seems better. When we do biology, no, suddenly helium-3 gets worse. But of course, that's also to be validated. And moving from helium into from the beam to the dream is actually, we do consider helium as a particle therapy option in the Baltics. This graph, I think you already saw in the beginning of the week by Professor Manjit. It's taken from the Analyte network, is the particle therapy center geography in Europe. And as we can see, there's this blank space in Europe, Balkans and the Baltics. As for the Balkans, it was outlined already in the Monday in the talk by Mariusz and Elena, there's the CEAST project. For Baltics, we would also want to do project, so we would have particle therapy centers, because sadly, the thing is that this blank area actually corresponds to sort of the same region in cancer mortality in Europe. So our options where we could have gone with particle therapy, of course, we could have just bought proton therapy treatment machine commercially, but we didn't want to do that. We wanted to do a new advanced facility with the possibility to do different ions and new treatment techniques. And of course, it's largely in collaboration with CERN, the ones that have the expertise, as we are quite new to the field in the Baltics. The NIMS idea on the helium synchrotron, the one that was outlined on Monday's presentation. So just a quick overview why we want to go into helium with therapy, because it's already considered also in some other centers, and HIT is already doing it, I think, is if you compare helium-4 over protons, there's the better physics characteristics, increased radiobiology, and probably, possibly a lower neutron dose. If you go to helium over carbon, of course, helium can't outperform the radiobiology aspects, but it has the lesser distal dose due to the decreased fragmentation. And according to my studies, it seems that it could be better suited for flash. So that's sort of where it stands against carbon. But the idea for the facility, it was also shown on Monday, the helium synchrotron. It is one of the technologies developed in the NIMS toolbox. And of course, we can't do it alone. And our idea is, of course, to collaborate with CERN very closely. And just few technical details is that, of course, it has the 3-block injector having different sections for helium-4 and protons, and a dedicated section for radioisotope production. The triangular lattice layout that was mentioned on Monday by Elena. And the idea is to have 3 beamlines, 2 treatment beamlines, 1 research beamline, and 1 of the treatment beamlines, of course, with the gantry. And the beam time, initially, we plan to do 50% of treatment with protons, and the other time we dedicate to research with helium. And finally, we would like, of course, in the end, to move in a full-scale treatment with helium-4. And the plan overall is also a bit mentions, of course, on the radioisotope production is why LINAC based, because of course, radioisotopes, they're mostly produced with cyclotrons now. Why we want to do LINAC based production is that it has lesser beam losses and activation if we do exotic production, not proton beams, but alphas or deuterons. If we have the radioisotopes, you can use them for diagnostical imaging, theranostics, or even targeted alpha therapies. And the isotopes of interest, of course, are the conventional fluorine and carbon. Then, of course, the main idea is to use astatine, because that's the project considered actually by the NIMS group. And we would also be willing to do any research in the community just in this field. Of course, it's not just a clinical facility, it's also intended to be a heavy research facility, mainly complementing the clinical activities, and also industry, because it would be the first infrastructure in the Baltics for the accelerator tech. And also we would like to involve all the relevant industries to construct this facility to so build the capacity in the Baltic states in this field. Plan, where are we? This is a bit of a political slide, so to say, so I'll try to be brief.
3 steps, just 3 more minutes.
Yeah, this is the last slide, so no worries. So like I said, the main thing is that this is one of the technologies developed in the NIMS toolbox. So the idea would be sort of that this center, together with CEAST, would be like a aldosterone canalus to PIMS. So we would be hopefully the sort of outcomes of the NIMS toolbox. And of course, this project, we would do it in close collaboration with CERN, as we are still building our capacity and our expertise in the accelerator field here in the Baltics. And the sort of political outlook of this, where are we with this, is that we've done a full concept paper on this. So the idea for the center, and the idea is of course supported by NIMS. It has received scientific community support within the Baltics and also political support within the region. And the next step is the main step to actually move forward to the medical community, because in the end, it's the main users of the facility. And the map on the left is, of course, we're not talking about where to build it now, but if we were to build it, it would be one of these 5 cities in the Baltic states, either in Estonia, either in Latvia, or even in Lithuania. So from me, that's it. That's my beam and my dream. Thank you for your attention, and happy to answer any questions if there are.
Yeah, thank you, Kristaps. Any questions for him?
It's also fine to go on, and if anybody has interest, Slack is always open.
Yes, Slack is always open for other questions, so don't hesitate. So thanks again, Kristaps. We'll be moving on to Rita from the University of Valencia. So, oh, there's one question from Celine. I have 1 more minute.
Yes, sorry.
Celine, go ahead.
Just maybe you said it in your talk. Sorry, I had shortly connection problems.
Yeah.
On what timescale are we talking? Like, you mentioned that it's a lot of politics and stuff, so I imagine it will take a long time.
Yes, it's of course, it's just we're at the initial stage, and I must say, of course, myself, I'm not a politician and in the end a physicist, but I think the initial talks are now that like we would like to move on if we get support from all the relevant communities in the Baltic states. The idea is to do the full project proposal and everything in the next call for EU projects, because of course we're going to apply for EU funding. So I suppose like more clear and like final ideas should be like in a year's or 2 years time.
Okay, so fingers crossed that it will take place. It sounds very cool.
Yeah, thank you.
Okay, thank you. Rita, so sorry for interrupting. You go ahead.
Can you hear me?
Yes, we can.
Can you see it?
Yes, go ahead.
Okay. So my name is Rita Viegas. I am a 3rd-year PhD student at University of Valencia. And at this Beams and Dreams, I will talk to you about my project project, which is a content camera for in vivo adherent therapy monitoring, which has the very funny name. So this is our research group. We'll have a pre-pandemic photographs. Some people are missing, but our group is called ARIS group, which means image reconstruction, instrumentation, and simulations for medical imaging applications, which is basically what we pretend to do. We belong to the, like I said, to the IFIC of Valencia, which is the Instituto de Física Corpuscular here in. So just a small introduction, although we already had a lot about photon therapy, so I'm not going to explain it, of course. I just wanted to make it clear that although we have a lot of advantages, we have also a lot of challenges, namely because of uncertainties. So besides the cost-wise, as the CNAO talk was very specific about this, we also have these uncertainties that we have seen, especially even this morning that we saw this talk about the in-beam pattern and so on to try to do the range verification. So we also propose something in the same way. So this is called the in vivo beam range verification. In vivo because we do it during treatment. In this way, we could try to reduce these uncertainties, and also we could reduce the margins that we need to do for the planning treatment following. Why is it not so easy as for example, in photon therapy? Because here, as you can see, I don't know, can you see my mouse?
Yes, we can see it.
So here we have the X-rays and the photons, and then we see that we have an exit. We can measure this exit dose and the entrance dose and have more or less a idea how much dose it was positive. For the protons, we don't have an exit dose, so we cannot measure directly the radiation. So what we want to do is to use secondary particles that are emitted due to the interaction of the ion beam with the tissue. Okay, so we can have different kinds of interactions. I will mainly do with this radiation, we call the front gamma rays, which are emitted within nanoseconds of the irradiation. And we have here also the 511 kV gamma rays, which are used, for example, in the PET. So in this time of treatment monitoring, what can we do? So we can do this real time, this in vivo monitoring through the use of this secondary radiation that is emitted very quickly. So we call it this 100 milliseconds after the irradiation. We can do it in the delay, which is with the in-beam PET. The main problem of the in-beam PET is that we will actually lose a lot of degrees from the photon beam that we can use to the gantry, and also the resolution of the PET must be so good. We can do a post-treatment PET where we move the table, but we have a PET in the same room, which is dedicated only for this purpose. It can take up to 10 minutes. And then we have this long after treatment, which is 10 to 20 minutes after, where we have a PET in the same facility, but it's not dedicated for this. And also we have then the long after therapy, an MRI, for example, after some days, weeks to see which damage was put on. The main problem of this, the green ones, let's say, and this gray one, is that we have body washout. So the longer it takes for us to try to measure where the interaction occurred, the harder it would be. So for that, we propose to use the prompt gammas. Okay, we have a continuous energy spectrum here. They are emitted, as I said, within nanoseconds after radiation. That's we call it in the in vivo, and we have a higher amount of it, higher than positron emitters. How do we propose then to measure? We want to measure their origin, the place where the proton beam interacted with the tissue. So for that, we propose to do content imaging. So we build a content camera where we have two detector planes. So in the first one, the gamma ray will suffer scattering. And in the second one, be absorbed or scattered again, and then we can actually have more detector planes. This is, I just put easier to show. What happens is that because we know the position of interaction in both planes, we can have the initial position of the front gamma, we can draw it on a cone, and then if we have several gamma rays, okay, we can then try to understand where it happened by the place where they join. So this example is with 5 events, meaning we detected 5 gamma rays which were emitted by one source, and here is 1000. So here we can clearly see the point. So for this, we developed the macaque sense for medical applications Compton camera. For a Compton camera to be successful in for treatment monitoring, besides a lot of more things, first of all, we need an excellent energy resolution under 4%. This at 662 kV, a very good spatial resolution around the 2 millimeter fluid half maximum, and a very good time resolution under 1 nanosecond. This because, as I mentioned, they are emitted within nanoseconds, and we want to be able to detect them. So these are the 3 versions we have had of macaque. What has been upgraded is the detectors themselves. So our detectors are scintillator crystals with silicon photomultipliers. Basically, what it does is that the radiation will hit the crystal, let's say like this, it will produce light, and this light will be measured by the silicon, and then the silicon will transform the light into electrical signal that we can measure. We have changed the crystals, we have changed we also changed the acquisition boards where we do the so the macaque 3, our last version, has a 5.2% energy resolution at 511 kV, more or less close to the 4% at 662. We have the spatial resolution under what we needed, so it's good, but we have here a big problem, as you can see, which is the time resolution of 20 nanoseconds. It is not feasible for clinical. So for that, they hired me. I'm developing this alternative macaque, which we call the macaque P, which makes use of this new technology, this new ASIC, which is a PET2 ASIC from PET's Electronics, which was developed mainly for PET scanners. But what it does is it allows me to do the charge and the time integration at the same time. On the other, we do the time. It's measured in another system, so we always have this problem of the time walk and so on. So here are my detectors. I mentioned the scintillator crystal, the lanthanol bromide, and a silicon photometer. Here we have both detectors and hardware, the readouts. Here is the newest version of my macaque. We have these cables that allow me to have the electronics further in the cases when we have that spin beam. So is it better? Well, energy resolution wise is not so better. It's the 6.4. We are still a bit far from the optimal value. However, time resolution wise, we are almost there. We are at 1.5 nanoseconds, which is, we're really happy about this. So once we test this in the lab and we saw that it worked and we had good results, we took it to in-beam tests. So we have this is a proton beam at the CNA Sevilla. This is an 18 MeV proton beam. This is an experimental beam. It's very clean. Okay. We have this graphite target, and here we have my prototype. This was when I was. Okay, my name is written out here. Here we can see what we did was we moved these targets by 1 millimeter in each direction. Okay, moving it, we are moving the position. Let's imagine if this was a tumor, we would be moving the tumor 1 millimeter to each side. So the position where I would measure it on my detectors change. And that's what we saw. So in this case, we measured 4 positions, 4 millimeters, 1 millimeter apart, I'm sorry. And here we have the 1, and this is 2 millimeters. So if we they are 2 millimeters. We also took it to clinical beams, which are not so clean. We have more. And in this case, it's at Quirón Salud in Madrid, where there is a synchrotron. And in this case, we put the both setups in the RW3 Phantom. And we also took it to Krakow last summer, where here it's already the updated version. And we did the same way, but this case we had a cyclotron. And with RW3 also Phantom, and then the system.
Rita,
Thank you.
three more minutes.
I'm finished. This is the last slide. This is our latest publication. I think maybe the slides will be uploaded, or if you want to see, I can send it to you. And okay, I forgot. And I'm finished.
Okay, Rita, thank you so much for your presentation. It was very interesting. So if you guys have any questions to ask her. Yeah, we have a question from Kyung-dong.
Hi, it was a very nice presentation.
Thank you.
Do you hear me?
Yes,
okay.
yes.
So I am working in a laboratory that also works for the PET imaging and PET development for Alzheimer's disease. So I am curious if you can say about your specification of the scintillator and SITM, if possible.
Yes, so our scintillator crystal is lantanobromide. It's a monolithic. And the silicon photomultipliers that I'm currently using are from HITR. So 25 micrometer cells, 64 cells.
Okay, so your SITM is from which company? Can you tell me?
They are now
okay.
they're commercializing through Broadcom.
Oh, okay.
We have also tried the Amamatsu ones, which we also like.
Okay, we are using Hamamatsu, and I was just curious.
We also have some Amamatsu.
Okay, thank you very much.
Results I dedicated.
Okay, thank you, Kyung-dong. Any more questions for Rita before we move on? Okay, so thank you so much, Rita, for your presentation. So we'll be moving on to Joseph Bateman, jointly from University of Oxford and CERN. So Joseph, the floor is yours. You can share your screen with us.
Okay, thank you very much. Share my screen. Right, is that in view for everyone? Okay, good. Thank you very much. So, as have you said, I'm Joe. I am a second-year PhD student currently in the John Adams Institute at the University of Oxford, and I'm also currently based at the CLEAR Accelerator at CERN. And I'll be talking to you today about my research on real-time dosimetry and beam monitoring for ultra-high dose rate, very high energy electron beams for flash. So, a little introduction to what VAGE is, the beam, and flash, the eventual dream. So I'm sure you probably already all know what flash is, but so I'll just briefly mention it again. The flash effect is the apparent healthy tissue sparing that is observed at dose rates is now being reported as above 40 grays per second, but there's obviously also many other factors that is now being realized that the flash effect could be dependent on, such as the instantaneous dose rate and other factors like that. In the graph here, we've got a few examples of where the flash effect has been observed for the dose pulse, the average dose rate as well. So the ones with the filled markers are where normal tissue sparing has been seen, and the hollow ones are where it hasn't. It's currently been demonstrated for low energy electrons, mostly, for example, the experiment at SHU, where they've done it on patients for protons, although this is mostly in transmission mode, and also occasionally photons, but obviously the photon production for flash is a lot harder. It has to either be through synchrotron radiation, since the fixed target flash with photons is a lot harder. Due to recent advancements in accelerator technology, it may now be possible to use very high energy electrons of 100 to 250 MeV to deliver flash to deep-seated tumors, which is not possible for the low energy electrons. Very high energy electrons also have many other advantages since they're electromatically charged, they can be steered, they can be focused, and they also have a ranging sort of dose distribution. So over here on the left, you can see the green one, which is the typical 200 MeV dose distribution for the electrons, or there's been some experiments to show they could potentially be focused to cause a sort of a electron peak, sort of a Gaussian here. But one of the major challenges for the dream of flash is the dosimetry for the ultra-high dose rate electron beams. So as you all know, ionization chambers, which are the sort of standard dosimeter for both reference dosimetry and for beam monitoring using transmission ionization chambers, is the fact that in the conditions for ultra-high dose rates, the response of these chambers, they saturate, as can be seen here on the right here. The collection efficiency drops significantly as the dose per pulse reaches the flash regions. Correction factors can be used to account for the decrease in this efficiency at the ultra-high dose rates, but as you can see here, the graph is on a steep gradient, so any uncertainty on here accounts for a large difference in the actual dose that the response is corresponding to. So this is really not ideal for when you're delivering all of the dose at once to the patient. Another issue with the transmission ICs is that the collection time is way too slow for if you wanted to use these sort of things of feedback for beam monitoring with flash therapy. So what are the requirements we're looking for an ultra-high dose rate dose monitor eventually for if we want to implement flash clinically? So obviously we want a response that does not saturate at these high dose rates. We also want a high temporal resolution so that we can potentially resolve the individual pulses or bunches, but also to be able to react quick enough to sort of cut off the beam once we reach the desired dose rate, prescribed dose. We also want a high spatial resolution to observe the beam size, the flatness, and the symmetry. We want a high level of beam transparency as well, so that the detector itself is not perturbing the beam before it hits the patient or scattering it too much. And again, we need a large area to cover the entire beam, especially for when we're using up to like 10 by 10 centimeter beams to cover the large tumors, particularly as VHE is going to be intended for the larger deep-seated tumors. So what are some potential options for the ultra-high dose rate dosimetry that's been presented currently? So recently, there have been modified ionization chamber geometries and designs by changing what gas is filling the region for the ionization. There's also recently been developed an ultra-thin plane parallel ion chamber that has been shown to actually have a non-saturating response in the flash region. Another thing is solid state detectors. So things like diamond detectors and silicon detectors have also been giving promising results into the dose rates required for flash. Scintillators, so scintillating screens and scintillating fibers as well, are known to have quite a quick response that can be potentially, once investigated, equivalent to the response and to the dose that is deposited by them. And also looking at accelerated beam instrumentation that's currently used in places like CERN, on the LHC, and other accelerators such as the current transformers and pickup monitors. So now onto the beam that could potentially make or break my dreams is the clear accelerator. So this is where I'm currently working on and also doing some of my experiments investigating potential options for detectors and instrumentation to be used in dosimetry. So it stands for the CERN Linear Electron Accelerator for Research. It delivers 60 to 220 MeV electrons. It's mostly a user-based facility and it's used for accelerator and component research and development, radiation to electronics, and also for medical applications, with currently a large amount of our work and research being dedicated to both VHE beams at normal dose rates and at potential ultra-high dose rates for flash. So we have the accelerator here, and on the bottom here we have the in-air test stand with what we call our C robot. So we're able to process many samples with films for dosimetry, but also other sort of biological samples. We have the water tank here, and you can also see the It's quite interesting on our camera there, the chunk of light emitted when we use our high intensity experiments of with the electron beam, with the Cherenkov being emitted over there. So some initial detector experiments that I've carried out at CLEAR to look at alternative options have included a PCVD diamond detector, a gemfoil detector, and a bare time-pick 3 ASIC without any silicon or gallium arsenide sensor, as obviously we're using a very high intensity electron beam here. These tests were looking at whether the response saturates at the sort of intensities that we needed. So we started at low beam intensities and slowly increased them up until we saw a saturation in the response. We also didn't go straight in with the high flash experiments because we did not want to damage these detectors, particularly the diamond one. Also looking at the sort of the temporal response of the detectors as well. So the initial results from these experiments was that the diamond detector, which was in fact employed currently as a beam loss detector at the LHC, saturated at an extremely low pulse charge corresponding to a dose rate of about 1.6 grays per second. So like I said, as it was used as a beam loss monster, we weren't so surprised at this very low saturation. The time-pick 3 chip also saturated at a very low pulse charge, equivalent to 5 grays per second. So again, would not be acceptable for the sort of flash responses that we are looking for. And the response of it can be seen here at 100 picocoulomb pulse and then a 250 picocoulomb pulse. This was also the bare time-pick chip, so this is without any sensor attached as well. However, the gemfoil that we used, all the way up to we used it up to 10 nanocoulombs per pulse at the time, and this corresponded to 200 grays per second. And this actually did not saturate. And you also can see a very good temporal resolution here, and we were able to observe the bunch spacing, which corresponded to the set values of the bunch spacing we were using on the accelerator. So some other sort of beam dosimetry we've been doing at CLEAR is looking at using the beam current transformer that was already installed at CLEAR just for giving a standard charge reading, which these transformers are able to do non-destructively through the sort of the magnetic field that is induced in the coil as the charge passes through. And we were looking at whether this can be used to provide non-destructive dose measurements. This has been employed in places like at SHUVE and in a lot of other preclinical low energy flash irradiation facilities, and where they have calibrated this to an ionization chamber. But here at CERN in CLEAR, we've been using film to as the reference dosimeter here to respond the response of the beam current transformer here. However, the issue we've been having well, not the issue we're having, the issue that is presented at CLEAR is that we don't have a flat beam, and it's also not a set size. We don't have a collimator. We have a varying beam size, and it's also in a Gaussian distribution as shown from the film measurement here. So we don't have the linear response that's seen in other clinics when we correspond the charge reading to the dose because of the varying beam size. So instead, we've been currently looking at correlating the response of the charger to the beam charge monitor to the film integrated area dose, or the dose area product. But another issue with this is that the beam size measurement is reliant on the fit. So we're having to investigate a more reliable fit on the sort of the Gaussian in order to have more reliable readings here to be able to sort of use this method for beam dosimetry at CLEAR. So looking at the next steps, so the existing range of detectors I did investigate, they were only applicable for the smaller beam sizes. So if we wanted to sort of scale this up for what a clinical range is, we'd have to use an array of detectors. As well as this, the BCTs would also be only sort of applicable for smaller beam sizes and not the large beams that we needed to treat the large sort of deep-seated tumors. The BCTs are also only capable of providing the total charge dose measurements as well. There is no information on the flatness, symmetry, or beam size on the beam. So these would only have to be coupled with other sort of measurements if they were to be looked at, employed clinically. We also I'm going to look at further tests on other solid state detectors, so such as different types of diamond detectors, because the ones we used were only sort of intended for low intensities, and also looking at things such as silicon carbide detectors, which are shown already in some preliminary tests to have a response linear up until the flash dose rates. I'm also hopefully soon going to be looking into characterizing and investigating a number of scintillating and optical fiber arrays. These have the advantage of the fact they can be scaled up to a large area, as shown here on the right. So in terms of the sort of size, they would be definitely suitable for a clinical application, but they're sort of the ones we'll be looking at are employed in the secondary beamlines at CERN, so not quite up to the intensities required, but we will be, again, carrying out similar initial tests to see what sort of intensity they can be carried up to and looking at their time response. And I'm also investigating sort of detectors and instrumentation for indirect measurements of the beam and for in vivo dosimetry to sort of complement some simulations that I've done previously. Thank you for listening. Are there any questions?
Thank you, Joe. Thank you for very interesting presentations. Any questions for Joe? Okay, so if you guys come up with any questions, DM him directly. You'll be happy to answer. So thank you, Joe.
Thank you very much.
We'll move on to the last presentation from Cameron Robertson, jointly from University of Oxford and CERN. So Cameron, the mic is yours.
Thanks very much. Okay, I think I can see this now. Okay, yeah, so hi, I'm Cameron. Like Joe, I'm also at the University of Oxford and CERN, and I'm also associated with the CLEAR Accelerator as well. But I'm going to be talking about slightly different end of VHE, which is looking at transfer lines rather than dosimetry. So I'll try not to repeat too much of what Joe's just said, because I think we've also got slightly limited time. So basically the same principles. Yeah, very high energy electrons. I've got my hair, I just realized I've got my hairpin the wrong way around there, but it's actually larger than 50 MeV, not smaller than 50 MeV electrons. They can be superior to conventional RT for treatment of deep-seated tumors. They can be more sort of inhomogeneous to, more resistant to inhomogeneities in the patient tissue. And unlike sort of low energy electrons, they can reach deep into the body, deep-seated tumors, so could potentially be used to be used for flash therapy. Another advantage of very high energy electrons is that they could potentially, because of developments in high gradient RF technology, be accelerated to say 100, 150, 200 MeV in a short linacs, not having to use a circular accelerator. And also because electrons are so much lighter than hadrons, a smaller required gantry would be required, or sort of could be required to carry out this treatment, since they are very much fully relativistic. So 200 MeV is basically their total energy and their kinetic energy. So the motivation of exactly what I'm doing is that for basically any clinically viable treatment machine for VHE, we would like to ideally get a flat beam. So what I mean when I say a flat beam is a beam with a constant transverse intensity distribution. So obviously, if you've got a beam that's got sort of big Gaussian tails on it, as would sort of somewhat naturally happen, you'd end up with a sort of lateral penumbra, and you might end up with an uneven dose distribution in the transverse plane. And then to sort of irradiate the whole tumor, you'd end up with increased dose to healthy tissues. So I'll probably refer to this as a flat beam, but when I say flat, I mean this constant transverse intensity distribution. So as well as trying to get this beam to be flat, we also would most likely want to magnify it, because after acceleration, a beam will be very small, a lot smaller than the size that a tumor could potentially be. So for the purposes of what I've been working on, I've been looking at magnification up to a 7.5 centimeter radius for a sort of single flat irradiation. Now, if this were to be used for scanning, you'd obviously require a much smaller beam. But for my current research, I've just been assuming this flat irradiation sort of maximum magnification that we would sort of reasonably expect. So yeah, to get these requirements, we use a transfer line. And there are basically two ways to sort of fulfill these requirements. So the first, which is the sort of standard one, which is what Marco actually discussed with us earlier, the sort of discussion of gantries. So normally it's basically just using optics, which basically we use a lattice or an arrangement of quadrupoles to magnify the beam. So I did some simulations to basically, with an initially flat beam, make a little lattice of quadrupoles with a so-called Riesenrad gantry. So if you remember, again, this was mentioned by Marco, a Riesenrad gantry is a gantry with a single dipole in it. And to allow the patient to be irradiated from 360 degrees, the patient is actually rotated around the beam, the dipole, which itself rotates. So it's a bit of a toy model, but it's basically just sort of prove that this magnification can sort of be carried out. And it was fairly simple to do, again, with an initially flat beam. So we've basically got two focusing quadrupoles, two defocusing quadrupoles of various strengths to carry this out in conjunction with this dipole. The issue with this is that it is very reliant on this initial beam distribution. So if you've got some jitter, if you've got some misalignments, we then do get the final beam being a little bit different. In addition to this, again, this is a standard problem with gantries with optics, is that you do have a very heavy system that's got to be rotated. Quadrupoles can be quite heavy. It is expensive, and when you're doing this rotation, again, it makes it even more likely you may have some sort of misalignment. So a second option to use is looking at, instead of just doing this magnification and flattening with optics, is instead using what's called dual scattering foil. So the basic principle of this is you've got your beam, which has got some sort of initial distribution. And in this case, it doesn't actually matter quite as much what the initial distribution is. You send it through a first scatterer, which is basically just sort of a flat piece of material. What this does is that does the sort of initial enlargement of the beam. However, this scattering is random. So you basically end up with a beam which has got a generally sort of Gaussian intensity distribution. What we then have is a second scatterer. Now that isn't a flat scatterer, so that scatterer is sort of shaped to complement the beam. So usually what we've been working with is looking at a second scatterer that's got a Gaussian shape to it. So as I say, it complements the beam. The center of the beam basically gets scattered more than the outside of the beam, and you end up sort of preferentially scattering the center to the outside, and you turn this Gaussian beam into a much flatter beam, as you can see in this diagram here. So you can see with one, big Gaussian, with two, it does some flattening, and then you could collimate the Gaussian tails off to get a sort of flat beam, but obviously trying to minimize the losses of the beam as much as possible. So what I've been working on is actually sort of verifying this to make sure this is something that we can sort of achieve with a very high energy electron beam. So we've been doing that at CLEAR, which is the same accelerator that Joe's mentioned. As he mentioned, it can go from sort of 60 to above 200 MeV, but I was just assuming 200 MeV just to compare the simulations. And at the end of the beamline, there's an in-air test stand where we can put various experiments in. So the purpose of my experiment was just to put one of these dual scattering foil systems. You can actually see, so first of all, in the top right, so that's one of the second scatters. You'll notice that it's obviously in a few sort of slices, but it is actually Gaussian shaped. And that was just 3D printed, just for ease, because you could always just load a 3D model and we could just, 3D print it. The first scatters were also 3D printed, but those were much simpler because they're just sort of flat cylinders. And we can, as you can see in this image here, this is an image of the inner test stand. We've got the C robot that Joe mentioned. So what we could have is we've got our first scatterers, which are down here, which could just be moved in front of the beam. And then the second scatterer was basically grabbed by the arm of the robot and inserted into the beam at an appropriate place to produce a flat beam. At the very end of this inner test stand is a YAG screen, which basically just produces light so you can see the sort of intensity distribution of the beam. So to actually carry out this design and sort of see what we were looking for, to basically see what we were looking for, to sort of compare simulations and what we had in CLEAR. We needed to set up a full Monte Carlo simulation of this. We basically used Topaz for this, which was mentioned by Alexander Gerbershagen yesterday. So Topaz is basically a wrapper for GANT4 to carry out Monte Carlo simulations. So it's quite simple. You can see that basically the clear beam is Gaussian, so without any scattering, we have an intensity distribution at the end of the beam line, which is a very sharply peaked Gaussian, as we'd expect. After the first scatter, so that's one of the flat scatters being put into the beamline, you get a much wider Gaussian. And then if we then insert the second scatter, the Gaussian scatter, at an appropriate place, we then get a much flatter beam, which could then be collimated. However, these are simulations. And as we all know, simulations and experiments don't always match up very well. Sometimes they do, sometimes they don't. Just a key point to notice here, so it's one of the drawbacks to digital scattering, is that if you send an electron beam through anything, obviously the final beam isn't just going to be electrons, there's going to be sort of contaminants, so you are going to end up with a quite significant number of X-rays in that final beam as well. So the first experimental test we did was just having the first scatter in and just seeing if that very simple single scatter matched up. So this is where we'd expect a Gaussian shape at the final Gaussian intensity distribution at the final screen. I mentioned we also 3D printed the first scatter. And basically the result of this was we had very good agreement between the experiment and the Monte Carlo simulation, which was set up to mimic it. You can see obviously there's slightly different intensities here, but when we did actually a Gaussian fit to it and looked at the fit parameters, we have an agreement of about between 1 and 2%, which we're very happy with. When we then expanded that, so we put in the second scatterer and did the full dual scattering foil experiment, you can also see that these are in the same situation where these actually do match up quite nicely as well. You will notice here, obviously, because we've got a flat distribution at the final YAG screen, a Gaussian fit doesn't actually describe it that well, but for this stage, it was sort of the best thing we could look at because you can do the same Gaussian fit to the experimental and the simulated data and see how it compares. So this is why you note the residuals isn't just sort of, you basically end up with this characteristic ring, which shows that when it's subtracted the Gaussian from a flat profile, you end up with this ring. And again, yes, we ended up with quite good agreement between, well, actually very good agreement between the experiment simulation, still only a few % difference in the fit parameters. A key point here is that in the simulation we're comparing with here is only electrons. And yet, I obviously mentioned that we end up with quite a lot of X-rays, and yet on the final screen, it does agree quite well with the simulation that was only the electrons. So what's causing that difference here? Well, the thing is, this YAG screen that we use at the end of the beamline appears to be much more sensitive to electrons in the final beam than it is to X-rays, because when we do the same simulation, but counting all the particles, so the X-rays and the electrons, we actually see that the final beam distribution is pretty much Gaussian because we've got very limited space in the inner test stands, which means that our scattering blocks are quite thick and we do end up with quite a high production of X-rays. But basically, yeah, as I said, we didn't actually see that in the experiment, which implies that yes, the screen is seeing basically almost entirely the electrons, and that the profile from the profile from the X-rays is being dominated by the electrons instead. So we've got another experiment planned to try and sort of pick apart some of these details. So that's basically looking at using aluminium scatters instead of a 3D printed one. The reason for that is that basically the 3D printed scatterers, while quite nice, we're actually able to machine out an aluminium scatter much more precisely. Aluminium obviously doesn't bend. We actually saw that when we were placing the second scatter into the beamline, it ended up sort of buckling a little bit under its own weight, and that will cause some small uncertainties. But as you saw, the agreement was still quite successful. In this future experiment as well, what we're going to try and do is look at, as I say, trying to pick apart how much of an effect this X-ray, these X-rays in the beam would actually have an effect on the dose itself. So we're thinking about using a dosimetric film, also a radiochromic film into the beam, because that we'd hope is going to be maybe sort of, it's basically proportional to the dose. So the electrons might not necessarily dominate, and if the electrons do still dominate, that maybe implies that the effect of the X-rays on the final dose might not be so significant, in which case this X-ray production might not be such a drawback to this sort of concept of using a dual scattering foil. So basically, I spent most of the time talking about the beam, but I think trying to sort of determine, characterize this X-ray distribution, how much of an effect, looking at the dose distributions of the total sort of scattered beam in comparison to sort of a pure VHE beam, that's going to be the sort of dream and the things that I'm going to be working on in the future. So that's everything. I'm obviously happy to take any questions.
Great, thank you so much, Cameron. We have one more minute for some questions you might have for him. I don't see anything in the chat, so
That's not a problem. I'm obviously always happy.
yeah.
If anyone wants to send any questions along. We're also going to have a couple of dedicated rooms in the spatial chat student session at the end, where we're happy to answer any more questions you have in any of our presentations.
Perfect. Okay, then thank you so much. And I guess I'll pass on back to Professor Manjit or Nicholas for the next section.
Thanks a lot, Sabiba. Thank you to all the presenters. Very nice. And so maybe we want to take a photograph before we go and pass on to Nicholas Hammond, because he's actually chairing this session this afternoon. Nicholas, what do you think about a photograph?
I didn't get you, Manjit.
Sorry, I was saying maybe we have a photograph just